slides?
Yes.
Yeah, we're good.
Perfect. And you're hearing me okay. All right. So thank you very much for the invitation today. I'll speak in English, but I'm happy to take questions in English or French afterwards. As Jean-Claude said, my name is Sébastien. I'm a research associate in the Michael Platt Lab. And today I will be talking about two things. First, I'll talk about the open optogenetic database that I've been piloting for about a year now. I'll end with a little bit of my own work in using optogenetics in non-human primates. So I don't need to explain what optogenetics is. I think most people are aware of it. But in a nutshell, optogenetics allows both inhibition and excitation of neural tissues as opposed to electrical stimulation, which stimulates the entire parenchyma. And it also affords cell type specific modulation using viral vectors and promoters that allow targeting genetic effectors to specific cell types. But the technique has been largely developed in smaller rodents, and therefore the technologies are adapted for the smaller rodent brain, which is a caveat when trying to use the same tools in the non-human primate. This is just a little overlook of the current technology, the current literature available. Here, if we have a look at all the papers that are being that are using optogenetics, starting for the first paper. I don't know if you can see my mouse cursor, but starting with the first paper in rodents, which was in 2007, and in non-human primates in 2009, and just seeing that there is a widespread adoption of optogenetics in rodents, almost an exponential growth, whereas in non-human primate we have a very slow adoption of the technique, which is a very linear increase that is not so impressive in comparison to rodents. And so that seems to indicate that there is a failure in translation. Of course, the monkey brain is different from a rodent brain in several different ways. The anatomy of it, if we're talking about volume, we're talking about a brain that is 200 times bigger than the mouse brain. The immune system is much more complicated and mount complex responses to foreign proteins and transgenes. And we also don't have widely available transgenic models that can facilitate some of our genetic approaches. So there are different issues here that need to be addressed in order to make optogenetic useful in non-human primates. So when I started my program here at Penn about 3 years ago, I wanted to launch optogenetics in non-human primate here, and I realized that there wasn't a lot of information out there in terms of the published literature, about around 50 publications at the time only. And so I went through several conferences, talked with investigators who have experience in optogenetics, and it came clear pretty quickly that a lot of the tests that are being conducted are never published, and that some of the people who have success and do publish articles had multiple failures beforehand. And often they decide to quit the field after publishing a single high impact publication. And so I was a little bit curious about that and discussed this with, at the time, James DiCarlo, who's the director of the McGovern Institute at MIT. And he told me, well, in our department, we have a little running list of all the tests that our investigators are conducting using optogenetics in primates. And so maybe you could use that. And I said, yeah, please give me this. And I will try to make it an international collaboration, listing all of the attempts that are conducted around the world using optogenetics in primates. And so I started with that and created a list trying to figure out who's doing optogenetics in primates, which in itself is a difficult job, starting with publications and then going to posters, and also word of mouth, asking investigators if they know of any other investigators who have tried it and maybe have never published because they only had failures. Creating that list of investigators and then just contacting them by email. I partnered with 3 well-respected NHP researchers in our field, James DiCarlo at MIT, Michael Platt, my supervisor here at UPenn, and Krishna Shnoy at Stanford University. And then just sent these invitation emails to the people I've identified, trying to state what the problem currently is and what we think could be a solution through a collaboration. And I'm just going to read, this is an excerpt of the actual email. This one was sent to Desaroth. I just say, currently optogenetic in NHP is performed with a trial and an error approach. with a large number of unsuccessful experiments remaining unpublished and unknown to our community. As a result, efforts are duplicated across labs, and experimental decisions are based on scant scientific evidence. And I don't want that for my own research program. I don't think anybody wants that either for their own. So can we build a resource that is going to allow us to make evidence-based decisions when designing an experiment in non-human primates using optogenetics? So I contacted around 66 labs through this initiative, and I was super surprised and happy that a vast majority of them were willing to contribute to this initiative. So 52 out of 66 said yes, would be happy to send some data. When some work was involved to actually send the data, about 45 of them actually did send data. And out of these 45 labs, we have 32 who have contributed unpublished data, things that were never known to the wider community. Here are the labs that have contributed to the initiative. In bolds are the labs that contributed also unpublished data. And you can see that this is a very international collaboration. We have 42 labs now represented in 7 different countries. And it was an amazing experience for me to get in contact with all these people and discuss different techniques that they're using and the problems that they have encountered. And to receive their data, and I've helped them to standardize this data and to put it into a database, which is a very simple Excel file. And you got a little snapshot of that Excel file here, whereby every column is a parameter of an experiment, going from the species of the animal to the viral construct that's used, all the way to the surgical technique and access, and the outcome measure. And every line is a different injection experiment. And so we have a whole bunch of them now. Through this initiative, we collected around 1,000 of these experiments. Around 500 of them had already been published, but the other half, 500, had never been published in the literature. So we're quite happy to double the size of the literature in that respect. As I said, around 45 labs contributed to this. And this is just to give you a sense of the effort of communication. This is something that really anybody can do with a laptop and a email client. And a little bit of time. And so just send a lot of email, help people out through their questions and so on, and eventually build this initiative. And so with that data, now we can start to have a look at a couple interesting trends. So let's just do descriptive statistics here and have a look at what people are actually using. So in terms of the species that people are using, we can see that the rhesus macaque is the most widely used animal model in optogenetics and primate optogenetics, with the marmoset at 6% also becoming strong. We can have a look at the different viral vectors that are being used. We can have a use at the genetic promoters, the reporter proteins, and the opsins that are being used. And then I asked the investigator to report all of these parameters, but also to tell me what outcome did they get out of this experiment? Like, did it work? And so this was kind of an open question. Investigators were offered to write some free texts beside every line of this database. And I received comments such as we got no expressions in cells, or we got strong neural response, or weak behavioral effect. And I needed to kind of find a way to quantify this so I can run some statistics on these outcome measures. And so I classified these comments in 3 main outcome measures, either through histology, the analysis of postmortem tissue, or in vivo tissue using fluorescence imaging, the physiology, so did the neuron respond to light stimulation, for example, and then behavioral effects. And behavioral effects can go from very simple eye movements generation to influencing a decision variable in a social decision-making experiment, for example. And then I have a look at the strength of the effect that's reported. Did they get no effect at all, or did they find a strong effect, or a weak or mixed effect of the manipulation, or did they not test it at all? And so classifying all these outcome measures, then I can have a look at the success rate of different experiments according to the category of outcome: physiology, histology, or behavior. And we can see that a large proportion of tests using physiology or histology report strong effects of optogenetics. And also a large proportion of them do not test either physiology or histology, and that's for a simple reason. In monkey business, usually we try to keep our animal alive as long as possible, and so people who do physiology usually don't sacrifice their animal and don't have a chance to look at histology. And there's also a large part of histological tests that are purely injection experiments that don't look at physiology, because that involves a lot more surgical techniques and time. But I think the important take-home message from this slide is the behavior. If we worry about making optogenetic a clinically meaningful tool, we need to have an impact on behavior. And the vast majority of tests either did not test behavior, and those who have had weak success, certainly not as representative as what would be indicated from the physiology. And so I think it's important to consider that right now, the problem that we need to address as a community is to adapt the technique such that we can generate behaviorally meaningful effects using optogenetics. So using these outcome measures, I can also have a look at each of the parameters, each column in that database, to try to see if there's any insight about possible best practices that we should use to make our experiments successful. Here, one choice that we have to make is what species of monkey should we use. And here we can see that marmosets and rhesus monkeys are the species that seem to yield the highest success rates in optogenetics. We can have a look at the viral vectors also. Here, I've got dotted bars and full bars because I have a statistical threshold here for power. So the full bars are viral vectors that have been tested in 5 different monkeys and in at least 20 different experiments. So this is kind of higher, more reliable, and then the ones that are dotted. So we can see, for example, AAV9 as one of the viral vector that leads to a lot of success. And I understand that this is not a final answer. I don't think everybody should use AAV9, but I hope that this is at least just a start to get some insight to people who are launching experiments about what they should use. Opsins, in terms of success, we can see NPHR3 as an inhibitory opsin that's very successful, and C1V1 redshifted excitatory that also yields good results. And I'm personally interested in surgery a lot, and so there are a bunch of information and insights about surgical considerations when a surgeon needs to make a decision about how to access the brain and deliver viral vectors to the brain. There are different ways to do that. You can do it through an open craniotomy and endotomy and directly visualize your needle penetrating the parenchyma, or you can use an implanted cranial chamber that provides access for a recording electrode, for example. A lot of monkey neurophysiologists already are familiar with these chamber implants. Or you can use more sophisticated techniques like artificial dura that provide both access and visual access to the cortex. People also have to decide, well, when I'm injecting, what parameters should I put on my pump? Should I inject very fast? How fast? How slow? How much should I inject? And here, some guidance about what most people are using. People are injecting at around 250 nanoliter per minute and injecting 2 to 3 microliters per site. And here, this is a bimodal distribution because we can see some tests that are conducted at very high injection rates and injection volumes, and these are the tests that use a convection enhanced delivery techniques that I'll be talking a little bit more when I'll talk about my work. We can have a look at the economy of all this. Where are people getting their vectors? We can see that University of North Carolina provides a bulk of the viral vectors. UPenn, where I am, is also a large provider, although I understand that right now they will scale that down and only provide to people locally. But they've deposited all their plasmids to Adgene. I've been ordering from Adgene. I have a very nice experience with them, both in terms of delays and prices. Very happy also getting my viruses from Stanford through a collaboration with Carl de Serath. So different options out there. So all of this data is an open science initiative. So everything is available on the Open Science Framework website. And you can access the database on this page, NHP Optogenetics Open Database, and you'll find a whole bunch of instructions about how to contribute and how to consult this database. And it's important for me to say that this is still an ongoing initiative. So if you want to contribute to this database, please do so. Reach out to me. You'll see my email address at the end on the last slide. Also, a lot of the data and the analysis were published last year in a paper in Neuron with the authors that have contributed to this initiative. I highly encourage people to go to the paper and have a look because there is in the supplementary material a section that I think is really a gold mine. When I wrote this paper, I asked every contributing lab to provide a 200-word paragraph about what would be your advice to someone who starts in the field. What do you wish you had known that you know now when you started? And so a whole bunch of advices about what to do and what not to do that can save you months to years of work when you start your own program. So using all of these insights from the database, I was able to start my own research program and having an idea about what needs to be addressed in primate optogenetics to make it work better and how to do it. So my objective, of course, is to optimize and develop an optogenetic platform that could eventually lead to clinical applications in human patients. And this is not a far-fetched idea. This is something that both Boyden and Desa wrote about some years ago, talking about the translational potential of optogenetics applications in neurology and psychiatry. Some companies like Circuit Therapeutics are trying to get into that business. And recently, a couple of weeks ago, we got a beautiful demonstration that it could work because it seems to work, at least in the eye. Paper in Nature Medicine published by both a French and Switzerland-based group that injected viral vectors in the eye and partially restored vision to a blind individual. So I think this is super encouraging results. But of course, accessing the eye is much easier than accessing the brain. And me specifically, I'm more interested in the cortex. And so I try to develop technologies and surgical techniques that address two of the main problems that I think we have in primate optogenetic. Problem number one is to develop surgical techniques that allow for large-scale transduction of neural tissue, as opposed to the techniques that are used right now in mouse that transduce about 1 millimeter cubic of cortex. We need to definitely scale that up. And then the second problem is large-scale illumination of the cortex. How do we bring light in an untraumatic, safe way to the brain such that we can have effect on large swaths of cortex that are more likely to lead to clinically meaningful behavioral effects? And so here on the left, you see a little bit the concept that I have in mind of a portable, wearable optogenetic brain stimulator that provides cell type specific modulation through excitation or inhibition. So I launched this collaboration here at Penn that involves many different partners. Platt Lab's at the center, but I have partners in neuroengineering, the Brian Litt Lab, who are specialists in material science. I have partners in mouse optogenetics to learn from the state of the art in optogenetics in rodents right now, the Geffen Lab. And then there's a whole part that addresses the immunology and the safety of all that. And that's collaboration with the Wilson Lab, who run the Penn gene therapy program. So I've launched right now a large series of optogenetics injection experiments in monkeys to try to optimize injection protocols and surgical techniques, contrasting, for example, conventional, convection-enhanced, and intrasystemic magna delivery approaches to deliver viruses to a large volume of the primate brain in a safe manner. Also playing with different viral vectors, and capsids, and titers to try to optimize all of that. So I use different types of surgical approaches. Some of them are MRI guided, especially when we try to reach subcortical targets, and optimizing convection enhanced delivery, developing reflux resistant cannula that are custom made. So CED, just in a nutshell here, is a technique that allows a larger scale distribution of the viral vector in the brain tissue by injecting at a high pressure using a reflux resistant cannula, such that we drive bulk flow of the interstitial fluid that leads to a more homogeneous expression of the virus in the brain, as opposed to a conventional injection that relies on passive distribution of the viral molecules. So these are techniques that are borrowed from gene therapy. Intrasystemic magna also is borrowed from gene therapy. So I think there's so much that we have to learn from this field. I'm able to map these injections in the MRI by mixing the viral solution with MRI contrast agents, in that case, manganese salt. So after my surgery, I bring the monkey to the MRI, image his brain, and then get an idea of whether I hit the target and how large the transduction is going to be. And then through postmortem examination, we can have a look here using epifluorescence imaging at the scale of the expression in the brain. And you can see these spots here, the yellow spots, these corresponds to expressions, and these are a single injection. So this is just one little single injection at the center of this that's done in less than half an hour. We get about a centimeter of expression, which is you know, many fold larger than what's obtained in the rodent brain. We also perform regular histology to have a look at the expression of the opsin in the brain tissue using immunofluorescence here. Zooming in and having a look at the distribution of the opsin expression within the cortex, having a look at the layer specificity here that we can see using CAM kinase promoters that are excitatory neuron specific. Zooming in also and paying attention to the ultrastructural localization of the opsin to make sure that it integrates the membrane so it can do its job. And of course, we're spending a lot of time in collaboration with professional pathologists to make sure that this expression is done in a safe way that doesn't trigger an immune response, that doesn't lead to toxicity or inflammation. So the pathologists that I work with H&E stain a lot, and we pay close attention to optimizing our viral vectors to make sure that we yield a large scale transduction without leading to neural loss or inflammation. Now, the second challenge that we try to address is large scale illumination. And this is the collaboration with neuroengineers that here at Penn, where we try to develop soft materials that are biocompatible, that can shine light in a non-traumatic manner on the surface of the cortex. So of course, all this is used in combination with ultra-sensitive opsins like Switcher++. And we pretty much lay this little micro LED array that has the texture and thickness of about Scotch tape and on the surface of the cortex. And the Scotch tape is embedded with very, very strong micro LEDs that can deliver light to the brain in a non-traumatic manner, laid really just on the surface. Nothing is implanted inside the cortex. And this is as thin as 70 microns. So it's very flexible, and we're running biocompatibility tests. We've got amazing results. I'm using some additional materials that prevent encapsulation of the implant within the dura mater over time. And we're very happy with what we're getting over there. Finally, all of this is in collaboration with a company, a neurophysiology company called BlackRock Microsystems that is based off Utah. BlackRock Microsystem pioneered the Utah array that is currently used in human patients. You can see here on the left Buzz, who's a paraplegic patient who's got 3 of these Utah arrays in his brain. So we're using the same type of technology here, the Seraport connector that is FDA approved, and we're wiring it to our micro LED. And with BlackRock, we're developing a head stage that is a version of their Seraplex W wireless head stage, such that it will have LED driving capability. So battery-powered current that can drive the micro LED array to then drive cell type specific modulation of the brain tissue. And of course, we're running all these tests in non-human primates right now in hope to eventually translate that to human neurological patients. So I want to thank a couple of people here. My principal investigator PI, Michael Platt. I want to thank specifically the Canada Benting Fellowship that allow me to come here and do everything I do. We're funding by the Charles Kaufman Foundation, and I also have fellowships from the Human Frontier Science Program. I want to say a big thank you to all the labs who contribute to the initiative. I want to reiterate that everyone is invited to contribute to this initiative and to consult it. If you want to, please contact me. Here is my email address at the bottom of this slide. So with that, I'll say thank you to everyone, and I'm happy to take any questions.
Wow. Yeah, we're blown away. Thank you very much, Sébastien, for a very enlightening talk. Yeah, sorry for the bad joke. Okay, so I guess we will hold on for questions for now. I think I see some questions being in the chat, so that's a good idea. We can maybe keep them in mind for now, and then ask them at the discussion at the end. So we'll jump right away to Keith, to Dr. Keith Murai. So I'm just going to do a quick introduction of Dr. Murai. So Keith wears many hats. So I won't have time to mention all those hats. But he's currently the director at McGill of the Center for Research in Neuroscience. He also is a leader of the Big Brain program at McGill. He's also a professor at the Department of Neurology and Neurosurgery. That's his main job. He's also the co-chair of one of the big CFREF that McGill has. He is also the incoming president of the Canadian Association for Neuroscience. Now, his research, I know Keith pretty well, but whenever I read all of the research that he's involved in, and I never seem to be able to keep track. So his research, he's focused on cellular complexity of heterogeneity for developing an adult brain. He's been working for a long time on basic mechanisms of plasticity underlying memory. And he's also has a big translational slant working on mouse models of Down syndrome, autism, and Alzheimer's disease. Now, because he wasn't busy enough, he decided to undertake quite a spectacular initiative to pretty much from the ground up establish a Marmoset program at McGill. And I think this is obviously what he will be talking to us about today. So Keith, it's all yours.
Great. Thank you very much, JC. Can everyone see my screen? And is it in presentation mode too and not presenter mode?
Yeah.
Okay, great. Thank you very much, JC, for the very nice introduction. And thank you, Mariev and Mario, for the invitation to present here today at the Global Resource Seminar Series. It's really fantastic to be able to attend and participate in these types of presentations. They're so helpful, very amazing in keeping the community linked together. Okay, let's see here. So today I'm going to tell you a little bit about the research or the our organization of a Marmoset initiative at McGill University, and I'm just going to say up front that this is something that is involving a really strong collaborative effort across various members of the McGill community, and coming from different institutions, different locations, buildings, centers. And really, it's been something that I've been really excited about and very proud about over the last many years, because everyone is coming together and getting behind this initiative to provide resources, expertise, and planning for the future. Let's see. Okay, so let's go ahead and get started. One of the amazing things about our field, right, is the multidimensionality of the brain and the space that we operate. We operate on spatial, temporal, topological, behavioral scales. And Sébastien gave a really exciting presentation on understanding some of this space and how we can understand, different aspects of neuronal physiology and imaging, and use it in a good way, to try to understand how the brain works, but also treat neurological conditions to help with the quality of life of individuals with, for example, injuries or disease. So really, it's incredible how we operate in the field of neuroscience and all these different levels, from the genetic to the cellular, molecular, all the way to the systems and behavior levels. Really exciting that we have all these aspects in our field of research, and the fact that we can come up with really good questions. But in order to answer these questions, we need to have a variety of different tools and technologies, right? We need to have expertise in these different domains. At the same time, we need to have adequate models that can allow us to test the questions, test the hypotheses, and come up with very interpretable results. And so, as we all know, no models of the brain and the central nervous system are perfect. And over many, many decades, we've learned, right, that there are different models that are really good at addressing certain scientific problems in our field. For example, the squid for the Hodgkin-Huxley action potential generation, synaptic transmission in the frog, aplesia for learning and memory, and say rodents for spatial navigation and things like that. And really, there have been really well-studied models that have helped answer some very fundamental questions in the field of neuroscience. An additional model that's been very helpful and actually revolutionized the field is genetic engineering in mice. And I've been working on genetically modified mice for over 20 years now. And really, this has changed the way we can study the brain and the nervous system and get at the fine details, right, of molecular mechanisms that underlie, say, how neurons develop, how they form connections, and how they might get sick and die over time. At the same time, we can use these methods to not only look at neurons, but also other cells such as oligodendrocytes, astrocytes, microglia, and how they participate in basic function of the brain, but also disease states. So rodents have been very helpful over the years in helping us to understand cellular and molecular pathways involved in brain development, function, and disease. But rodent models in particular have had some challenges over the years in terms of their translational capacity. Many models of disease have been created in rodents and been tested, and many research projects have been able to solve disease states in rodents, for example. But there are some problems in terms of translation of some of these findings to creation of new therapies. And so what are the reasons behind this kind of struggle to translate findings? Well, there are some strong differences, of course, in brain anatomy, cellular complexity, and function between rodents and humans, and we all can recognize this. At the same time, there are disparities in the type of circuits that might underlie cognition behavior. And what has happened, though, is that there are many preclinical models in mice in the field of neuroscience, but when these results get translated to clinical trials, there are some challenges with respect to efficiency and strength of these therapies in human trials. And there's a lot of reasons for why this is the case, including the clinical populations that are tested and the intervention points and things like that. But in general, there have been some struggles with rodent models, preclinical models, and translational success in human trials. So there's definitely a need for additional models that can help us understand certain aspects of central nervous system function, and in particular deal with brain disorders and diseases. And so, I just pulled up a couple of examples here of how, there are some very fundamental differences between cells in our brain and those in other species, for example, in rodents. On the top here is just an example of a layer 2-3 neuron in mouse and in human. And this is taken from a paper in Neuron recently, a few years ago. And as you can see here, just looking at these layer 2-3 neurons, there are some really interesting differences in terms of the size of the cells, how they elaborate and grow, and how they interconnect. But at the same time, there are molecular changes and differences in terms of what kind of channels these neurons express. And this particular study looked at the HCN family of channels and looking at these hyperpolarizing mixed cation currents, and found some really fundamental differences in the types of channels that human neurons express and utilize for their physiology in comparison to what's known in the rodent. So there's some interesting added complexity to human neurons with respect to their anatomy and physiology. And this could have an impact in terms of trying to translate findings between rodent and human, and understand also disease states of these two cells and how to intervene. So this is the neuronal side. There's a whole other side to it also with respect to the glial cells. People usually think about glial cells as being fairly uniform cell population that's involved in kind of brain homeostasis. And indeed, that's true. But these cells also have very specialized functions. And I'm looking here at the astrocytes, where you can see actually some very interesting differences in terms of the size and scale of, for example, of the mouse astrocyte versus the human astrocyte, in terms of number of processes, cell volume, number of synapses that a single cell can associate with. So some really interesting differences between glial cells in the brain and how they might function in terms of cellular communication, physiology, and involvement in overall brain function. So, there are definitely some limitations with the current animal models that we are studying, and for the most part, the vast majority of researchers utilize the rodent models. But really, we have some challenges in terms of kind of translating these findings from rodent to human, and not only in terms of understanding fundamental properties of the brain, but also translational aspects, developing new therapies for mental health, mental disease, and neurological conditions. So really, we need to be thinking about our models. We need to be thinking about what kind of questions we're asking and how far maybe the models may be able to take us. And in some cases, we might need to think about additional models that need to be incorporated and to answer certain questions. And so as part of the McGill strategic research plan is to understand the potential of the human brain. And we want to use certain models like rodents to try to get there, but there are some limitations. At the same time, part of the McGill plan, strategic research plan, is to facilitate the prevention and treatment of mental and neurological disorders. So really there's this push at McGill to try to understand the human condition in a much more effective way. So one platform of research that we've been developing over the years at McGill is the Marmoset model. And I'm just going to give a quick primer on the Marmoset model and why this model might be useful for neuroscience research now and in the future. So what are marmosets? Marmosets are a small New World monkey. They're native to South America and originated from the northeastern part of Brazil. And they're fairly small animals, about the size of a squirrel and about 250 to 350 grams in weight. Marmosets have very complex behaviors in terms of their vision, language processing, motor, sensory, cognitive, and social skills. They're an interesting species in that they show pro-social behavior. So they live together as family units. Males and females tend to pair bond and have cooperative raising of their young. And this interaction between the parent and the babies and the young is really important for development of cognition and behavior, for example, in terms of vocalization behavior. What kind of sets aside marmosets, what makes marmosets interesting as a model also is their fairly short reproductive cycle. Their reproductive cycle is about 6 months, and marmosets females give birth to either twins or triplets. So they reproduce quite quickly, and that could be an advantage in the future, and I'll talk about more why that might be. So what about the strengths of the marmoset model for neuroscience research? Well, there are some definitely interesting aspects of the marmoset model with respect to their types of behaviors that they show. And the fact that these animals are very social, they like to interact with one another. So the marmoset model really could be an important model for social cognition and understanding how individuals interact over time in their environment. And also for certain types of understanding of certain types of pattern behavior, for example, language development, vision, and other types of sensory integration. What about their brain anatomy? Well, as Sébastien showed some amazing pictures of the macaque brain and the surgery going on there, the marmoset brain is not like the macaque brain or the human brain in terms of its kind of level of complexity. It doesn't have all the sulci and gyri to give this kind of anatomical complexity at kind of the face value. But marmosets do have a quite complex brain. As you can see here on the left, marmosets have a brain that's a bit larger than the size of a rat brain, but quite smaller than a macaque, and of course the human brain. And a noticeable feature of the marmoset brain, though, is the fact that it's smooth. It's lissencephalic. And so it doesn't have a lot of the kind of overall architecture of the brain of macaques or humans. And this is interesting because it could be used as an advantage in some forms of imaging experimentation. The marmoset brain is, although it may not have some level of anatomical complexity, it has some very interesting complexity in terms of specialization, though. And on the lower right here is just looking at the areas related to the prefrontal cortex. As you can see here, this is the rodent prefrontal cortex and the basic organization and relationship, say, to the human prefrontal cortex. As you can see, the rodent cortex is organized in a quite different way and has not as much representation. The marmoset prefrontal cortex is much more developed, and researchers have been able to go in and try to understand how it might be compartmentalized in a different way and be more relatable, say, to the human and, say, macaque prefrontal cortex. As Sébastien mentioned, marmosets also have a very developed blood-brain barrier, immune system, have interesting pharmacokinetic properties that more closely mimic human properties. They have interesting sleep patterns and overall behave more similar to humans than, say, to rodents. Another interesting facet about marmosets is the fact that they do have actually some naturally occurring pathologies in the brain that, say, rodents don't have. For example, accumulation of beta amyloid into plaques. And marmosets aren't known to have Alzheimer's disease, but they do have some of the cellular signatures of neurodegenerative disorders, for example, beta amyloid plaques and some aspects of neurofibrillary tangles. The fact that they have these naturally occurring pathologies suggests that these animals might be susceptible to the same kind of mechanisms of disease as in humans. As I mentioned earlier, breeding and reproduction of marmosets is fairly rapid. They give birth twice per year, and usually to 2 or 3 animals. And marmosets can become sexually mature by about 1.5 years of age. So across internationally, there are becoming more resources available to understand the marmoset brain and ways of working with marmosets as an experimental model. In terms of resources available, we now have the marmoset sequences, DNA sequence. There are anatomical and molecular atlases. I'm pointing up to this one here is the marmoset brain in stereotaxic coordinates that was done by Paxinos and colleagues, including Michael Petrides at the MNI. And there's also there's a marmoset gene atlases, for example, from Riken in Japan. Tomoe Shimogori has done an amazing job of trying to create an in situ hybridization atlas of the marmoset brain. And there's some really interesting things that are coming out of this platform in terms of looking at differences, say, in the molecular expression of genes in rodents and in marmosets. And of course, one really interesting facet about marmosets is the ability to do genetic engineering, the ability to insert genetic reporters, but also, for example, genes that carry mutations into marmosets, and to use technologies such as CRISPR-Cas9 and prime editing and other types of new technologies to introduce genes into the marmoset and to potentially create models of disease. And in fact, a lot of our Japanese colleagues have done this, including making models of Parkinson's disease, Alzheimer's disease, and ASD. And there's been really a remarkable investment in the marmoset model over the last decade. CIA in Japan's, doing an amazing job of investing resources into this as part of their Brain Minds initiative. A large part of this is Erica Sasaki's work at the Central Institute for Experimental Animals, where they're leading a major effort to create genetic models of disease using the marmoset model. MIT is also leading the way in terms of CRISPR-Cas9 mediated genetic manipulation in the marmoset to create specific point mutations in genes that are, say, related to neurodevelopmental disorders and autism. University of Pittsburgh, the German Primate Center, as well as NIH have also created models, genetic models in marmosets. So I just pulled up here a couple of papers that have really started to transform the field of, transform the field of genetic engineering in marmosets. There was a really landmark paper that occurred in, that happened in 2009. It was a paper published in Nature from Erica Sasaki. And this was the first evidence that genetic engineering could take place in marmoset embryos to create certain models. And really this changed, dramatically changed the way people viewed the marmoset model, from going from a really nice model of physiology and behavior to potentially understanding maybe disease states using this model with genetics. Since this time, there have been a lot of other papers. I'm just showing a few here. This paper is from NIH and Alfonso Silva's group, inserting GCAMPs into the marmoset brain. This is from the German Primate Center, also inserting GFP into the marmoset brain. And more recently, Erica Sasaki's group at CIA has created the first familial AD model by inserting mutation that deletes exon 9 of the presenilin 1 gene to create this model of familial Alzheimer's disease. So technologies have now been developed to basically go in and modify the marmoset genome and to create different models. So marmosets also, it's a great model in terms of for many different reasons, but there are some major challenges when implementing the marmoset model. And I just wanted to quickly go over those in case some of you, may be interested in implementing the marmoset model at your institution or research center. And so one important thing to keep in mind about marmosets is that they are a non-human primate species and very precious animal species. And really research with marmosets needs to be well justified, just like other animal models that we use. But in particular, we need to think about how the marmoset model can tell us or answer certain questions that maybe other models can't use. For example, human tissue models or iPS cells and things like that. Or for example, rodent models as well. And marmosets do require very specific infrastructure, logistical considerations, and also very specialized veterinary care. Animals, animal facilities need to be able to handle non-human primate species. So marmosets are handled in similar ways to macaque and other species in terms of health monitoring, in terms of intervention points and things like that. So this needs to be taken into account. There are also some logistical constraints with the marmoset species with respect to how they're housed. Generally, they're housed in family units where you can have a mother and a father, and then up to 2 generations of offspring with them. So as you can imagine, marmosets need a lot of space, and they're in large enclosures, and they love to live in high places. So you got to take that into account too. You can't. You need to have enclosures that can accommodate some of their more natural behaviors. Expertise, this is another thing. Marmosets are not like rodents. You need to handle them, like non-human primate species and control their reproduction, control their diet. They need a very enriched diet in order to stay healthy. And luckily, marmosets, they don't carry herpes V virus, which can be fatal for humans, but they are a very susceptible species to disease, in particular they're susceptible to the measles virus. And so they really need a close interaction with veterinary staff to maintain marmosets and to use marmosets for experimental purposes. And one thing kind of unique to marmosets is that they can be a fragile species in terms of showing some specific diseases such as marmoset wasting syndrome, where the marmosets will lose muscle mass and change their feeding behavior, and within a couple of weeks could actually, if this condition could be fatal. So generally this is not a problem for sites that use marmosets and take good care of the marmosets. So there's you don't commonly see this type of thing, but it can happen. One current bottleneck for marmoset research is the high cost. And when we first started creating some of our colonies at McGill, the animal cost was extremely high, $8,000 per marmoset. And you can imagine that's cost prohibitive for using the model for most researchers. And they're also very limited. And so they're not only expensive, but hard to get and could be hard to transport as well. So this is a bottleneck for marmoset research at the moment. And one last thing about marmosets, certain types of studies involving aging, they're not going to be, these could take time. So marmosets can live over 10 years of age. And so if you're looking at, for example, marmoset models of Alzheimer's disease, for example, the one that was created by Erica Sasaki's group, you may need to wait 5 to 7 years to see Alzheimer's disease phenotypes emerge. So it's not like in a rodent model where you can see disease phenotypes emerge within 6 months to a year. So just something to consider. So careful planning and coordination is really required. So the last part of the talk, I just want to focus on what we're doing at McGill to implement the marmoset model. So I spoke to you a lot about what the marmoset model is and what are some of the advantages, but also constraints and challenges to the model. So taking all this into account, McGill decided many years ago to create an integrated initiative where we have researchers from across the entire campus at both the main university, but also at the hospital-based research institutes, work together to develop the marmoset model and its use for neuroscience research. So several years ago, we established a kind of a consortium called the McGill Marmoset Innovation Corps, otherwise known as MIMIC. And MIMIC was built on the foundation of the fact that we've had marmosets at McGill for over 20 years now. And so a solid understanding of how the model works, how to take care of the model, and how to use it effectively. But really, much of that expertise and knowledge was consolidated in one location. So we decided to create this network within the universities so that we could maximize our resources, combine our expertise, and just try to eliminate as much as possible any waste and redundancy in terms of the experimentation that might take place. And this is also part of a strategic growth plan in terms of implementing new infrastructure, training people who are interested in using the marmoset model, and building know-how, and also coordinating recruitment. This is another important thing. It's oftentimes if you want to bring in a new generation of researchers that can study a certain area or field, and in this case, maybe a marmoset model. So we've been coordinating recruitment across the university too, to make sure we can get positions to bring in new researchers who actually utilize the marmoset model and have certain expertise. We have a scientific advisor committee that helps with oversight and utilization of resources. They help to prioritize the research and the deployment of the model, and really with the idea that you want to be able to use the model, but at the same time, you want to sustain the model and make sure that we have enough animals and utility for the future. Another important added feature of this Marmoset Innovation Corps was to promote collaboration. This is really important. And I think it goes hand in hand with what Sébastien was talking about in terms of getting people talking together, sharing data, sharing resources and insight into the use of a particular model or technology. And this has been extremely helpful because we've been able to not only consolidate effort at McGill, but also reach out in a unified voice, for example, to partner sites. For example, Erika Sasaki in CIEA Japan and Guping Feng at MIT and others. And we get to actually interact as a unified voice rather than a group of individual researchers that are working with the model. And really, this is important for very practical reasons. For example, animal husbandry, best practices for health for the marmosets and refinement to procedures, but also the sharing of tools, models, protocols, and in the future, data sets. And in the future, also coordinating the genetics of our marmosets to understand where they come from originally and how they relate to the marmosets that exist at other sites around the world. So how is MIMIC organized? Currently, we have 4 main MIMIC sites at McGill. This is on the main campus. We have the McIntyre Medical Sciences building. This is where we have a large experimental colony for marmosets, over 200 animals there, and where the animals are being bred for experimental purposes and for behavioral testing and where a lot of surgeries take place. You have the Montreal Neurological Institute, which is also a location performing marmoset research, and in particular brain imaging using MRI and PET imaging in the marmoset model. You have the Douglas Hospital, which hasn't started their marmoset research, but is interested in getting involved and developing, translating some technologies from rodent imaging, for example, using miniscope imaging in rodents, and using that technology in marmoset species. And finally, at the Montreal General Hospital, over the last several years, we've been building the marmoset transgenic core that's basically going to help supply marmosets that have genetic reporters, but also disease models in marmosets to these collaborating partners within the university. And I've been actively involved in this process, and it's been really an exciting initiative to get involved in. But it's very involved and very expensive and very time consuming, but it's been really rewarding, and we're actually quite close to generating some models in the coming year or two. We're just optimizing some reproductive technologies that are going to improve efficiency of model creation. So we're just on the doorstep of creating new models. So how MIMIC is supposed to work is that we have 2 major colonies at marmoset colonies at McGill. We have the McIntyre Medical Sciences Building colony. That's the main experimental colony that's going to provide animals to partner sites within the university, but also to collaborators. And then you have the transgenic core, which is also going to supply animals, but these will be genetically modified animals. But the idea is that we have some crosstalk and interaction and complementarity in terms of how marmosets and other resources are being provided. And importantly, we want to be tethered into the international community, but also our local community within Montreal, Quebec, and Canada in general, in terms of how we are performing marmoset research, but also providing maybe marmosets to partner institutions. And so this is part of an integrated plan to, like I said, to have complementarity and to reduce redundancy and increase efficiency. So, there's a lot of things happening with the marmoset model, both at McGill, but also internationally. And there's this growing set of resources available to use marmosets for neuroscience research. As I mentioned earlier, there's new genome editing technologies being developed that have been successful in marmosets, for example, CRISPR-Cas9, but also lentiviral approaches for creating models, as well as new optimized or optimize new imaging and physiological technologies for work with marmosets, including MRI, PET, 2-photon imaging, and physiological arrays that can be used as well. Behavioral analysis is something that has always been well developed in the marmoset community because they're an excellent model to understand, for example, social interactions. And I pulled out a couple of examples here, recent examples of using marmosets to understand social interactions and behavior. This is from Stefan Everling's lab that was published just recently in eLife, looking at how marmosets actually respond to different visual scenarios. For example, marmosets interacting in different ways on a screen, and they're looking at the brain of a marmoset that's observing certain types of social behavior and the activation of certain brain regions. This is an interesting study that is also, another interesting study is one that's in bioRxiv from Corey Miller's group in terms of understanding how marmosets might solve this, the cocktail party problem, where you have multiple auditory inputs coming in and conversations happening, and how an individual might be able to sort out the information coming in and hone in on certain information and have an effective kind of communication. And marmosets are very vocal animals, and they have all different types of calls and vocalization patterns, but are very good at actually deciphering communication between other animals. And so this is a nice study, a very interesting study in bioRxiv, using the marmoset model to understand kind of social behavior, communication behavior. And it's believed that humans might be using the same type of brain circuitry and some computations to solve this cocktail party problem. And of course, there are some new tools coming online. Mariev can talk maybe a little bit more about this in the discussion section, but including the development of 8 new AAVs that can effectively transduce cells in the marmoset brain. And new capsids are currently being developed globally that will work in marmoset brain to target certain cell types, but also create more widespread transduction across the whole marmoset brain. And this is something very similar to what Sébastien was talking about, as well as development of new technologies, for example, MRI and CT guidance of injections to increase precision and efficiency of the experimentation that's going on. Okay, so that was my last slide. I just want to, in terms of acknowledgements, just thank all the researchers, administrators, veterinarians, and technical staff, as well as collaborators that have contributed their knowledge feedback to this Marmoset initiative at McGill, and also for the just high level of collaboration. I've been at McGill for 18 years now, and for me, this is one of the first major examples of where you have different people, different researchers and community members across the entire university come together and put in financial resources, infrastructure, and expertise to try to get this model working and to understand the value of collaboration. I just want to thank everyone for their contributions, and I'm happy to answer any questions.